We all vividly remember the emotions stirred in us by watching Steven Spielberg's iconic film, Jurassic Park. The idea of recreating the flora and fauna of the dinosaur era sent fans of these giants into a state of indescribable excitement. However, what intrigued viewers even more was the straightforward solution to the problem of cloning dinosaurs. To the average person, the method described in the film seemed quite scientific. Modern science has no doubt that dinosaurs were warm-blooded, so the notion of preserving their DNA in a drop of blood found in a mosquito trapped in amber appeared to be a reasonable hypothesis. Unfortunately, even in a piece of amber, the structure of DNA would be completely degraded over such a long period, making it impossible to recover the encoded genetic information using any contemporary methods. Even the scientific approach to reconstructing lost genetic codes through the introduction of genes from modern animals, as depicted in the film, does not hold up under serious scientific scrutiny. While scientists involved in cloning and genetic engineering do implant nuclei from ancient organisms into the eggs of modern animals, only species that are genetically very similar can be used for such experiments. Thus, for successful dinosaur cloning, it is not enough to simply obtain quality genetic material and decode their genomes. The success of such experiments will depend on the choice of a surrogate mother for the new generation of giant reptiles. Furthermore, scientists are not aiming to resurrect every species that ever populated the Earth, despite their popularity and the public's preferences. One of the key criteria when selecting animal species for revival is their potential benefit to modern nature and humanity. Additionally, the conscious animal must be placed in a suitable habitat or seamlessly integrated into contemporary ecosystems. Therefore, for the successful cloning of extinct animals, high-quality genetic material, an appropriate surrogate, and comfortable living conditions are essential. As a result, modern researchers prioritize species that went extinct in relatively recent history over the dinosaurs. Currently, various research groups around the world are working on the revival of extinct plants and animals from different eras. Among these studies, several realistic and even successful projects stand out. For instance, recreating plants that thrived on our planet thousands of years ago is significantly easier. This requires finding seeds that, for various reasons, have survived in conditions that allow them to remain viable. For instance, a group of Israeli scientists successfully grew several date palms from seeds that miraculously survived, found in an ancient stone fortress. Out of hundreds of discovered seeds, 34 of the best preserved samples were selected, and only 6 managed to germinate. Each tree was named after a biblical character, and the researchers hope to delight humanity with the first harvest of dates that are approximately 2,000 years old. The scientists believe that the fruits from these palms will be significantly larger than any modern dates. In their experiments, biologists from Russia used seeds of a plant called narrow-leafed cotton grass, discovered in layers of permafrost at a depth of 40 meters. This perennial herb thrived in the Kolyma region around 30,000 years ago, and its descendants are still growing there today, thanks to the low temperatures. The plant cells were preserved well enough for the researchers to extract material for further cultivation. Initially, the cells were placed in a test tube, and under the influence of hormones, the dividing cell mass produced the first roots. Then, exposed to light, the plant sprouted its first shoots and leaves. This experiment demonstrated that living tissues can be preserved for extended periods at ultra-low temperatures and then revived. Of course, this assertion has only been tested on plants so far, but there are already several cryogenic facilities in operation for humans and animals. In these facilities, the bodies of individuals who hope for advancements in this field in the future are frozen. They or their relatives consented and paid significant sums to have incurably ill or deceased loved ones, as well as pets, preserved for up to 100 years. The last hope for these volunteers lies in their faith in medical technology progress that may one day save their relatives. However, an effective method for thawing patients has yet to be developed. One of the most intriguing studies involves the revival of a living creature that is just as popular as dinosaurs, the woolly mammoth. These furry elephants existed long after the age of dinosaurs, which gives scientists a better chance of successfully cloning them. Numerous well-preserved remains have been found in the permafrost, and their distant relatives, modern Indian elephants, could serve as embryo carriers. Although attempts to clone mammoths or genetically modify elephants to express traits of their ancient ancestors have not yet been successful, there has been an ongoing project for several years aimed at recreating the habitats of mammoths, saber-toothed cats, woolly rhinoceroses, and other Ice Age fauna. Interestingly, researchers believe that reviving the saber-toothed cat will be significantly easier than bringing back the mammoth, as modern African lions are genetically very close to them. Moreover, well-preserved bodies of these animals were discovered a few years ago. More successful experiments in this field have focused on reviving species that went extinct in the last 2 to 3 centuries. Scientists have made significant strides in cloning species like the passenger pigeon, which was driven to extinction just over a century ago, the dodo bird, which was wiped out by colonists on the island of Mauritius in the late 17th century, and the thylacine, or Tasmanian tiger, which disappeared in the first half of the 20th century. When it comes to reviving fauna from the dinosaur era, the challenges are much greater, as cloning these giants remains within the realm of bold science fiction. Researchers can only continue experimenting with the genes of their descendants, which modern science claims are represented by birds. For several years now, a team of scientists led by paleontologist Jack Horner has been attempting to create dinosaurs from chicken embryos by stimulating the expression of dinosaur-like traits during embryonic development. These traits include a long tail, a specific head shape, and the presence of teeth. The scientist claims that they have already managed to produce an embryo with a head shape and teeth resembling those of dinosaurs. He believes that within 5 to 10 years, he will be able to present the first revived reptiles that lived during the Paleozoic era. While it may still be too early to talk about the complete restoration of all dinosaur species in the foreseeable future, science is advancing rapidly. Perhaps one day, people will be able to see at least some dinosaurs in zoos or reserves, just as depicted in Steven Spielberg's films or Arthur Conan Doyle's books.